Good afternoon. I am Dr. Lauren Hughes, and I am very excited to share an overview of the work I have been doing to better understand the genetics underlying Pituitary Pars Intermedia Dysfunction in horses. This neurodegenerative disease, also known as equine Cushing syndrome, affects up to 15 to 30% of horses above the age of 15. The disease occurs when a portion of the pituitary gland, located at the base of the brain, overgrows. This overgrowth is due to a lack of dopamine activity and leads to excessive levels of certain hormones within the horse. These excess hormones contribute to a variety of clinical signs that can significantly affect the health of horses, including a long, shaggy hair coat, loss of muscling, change in behavior, recurrent infections, and laminitis, a painful condition that affects the hoof, causing loss of use and even sometimes loss of life. A challenge currently facing veterinarians like myself is that this disease can be difficult to accurately diagnose, especially in early stages, leading to many unregulated horses that become much more difficult to treat once the disease is identified. Interestingly, recent work has revealed that the changes occurring at the tissue level in horses affected with PPID are similar to some of the changes that occur in humans affected with Parkinson's, like famous actor Michael J. Fox. This has presented an exciting opportunity to now better understand the genetics of this condition in horses, as well as potentially improve the way we screen, diagnose, or even treat. Additionally, the work we are doing in horses may have the ability to translate back to human medicine and increase what we know about Parkinson's. Many diseases are a combination of the genetics an individual has and the effects of their environment. The goal of this project is to better understand the genetic aspect of PPID in horses by identifying genetic mutations that contribute to an increased risk for development of this disease. To address this goal, we obtained DNA samples from 36 horses, 23 of which were affected by PPID, and 13 that were deemed healthy, normal horses not affected by the disease. This DNA was then whole genome sequenced to allow identification of mutations or areas within the DNA that are different. Whole genome sequencing is a process that allows us to take a DNA sample from an animal, normally in the form of blood or hair roots, and determine the entire genetic code. Like is shown in this graphic, the DNA is first isolated, then broken down into small pieces, and assembled into a sequence through a multitude of steps. This sequence is then compared against a reference to identify mutations. At this time, we are still in the early stages, but have already identified over 130 genes that may contribute to PPID through their role in biologic processes or their relationship to diseases like Parkinson's. Closer evaluation of these genes has revealed over 80,000 mutations, some of which are high or moderate impact. This means they have a larger effect on protein coding and potentially functions of the body. Each of these individual mutations is then evaluated to determine if the mutation is real and assess its frequency in diseased horses. One concept that is very important to understand here is that each individual animal, or even all of us as humans, have millions of mutations present within our DNA that may not cause disease. This is why it is so important to actually compare the mutations identified between affected horses and healthy normal horses. Since we believe PPID is a complex genetic disease, multiple mutations are likely working together to increase the risk of development. In going forward with this project over the next few years, we will continue to expand the number of genes and mutations evaluated and eventually reassess the identified mutations in a larger group of PPID positive and healthy control horses. For many of us like myself, horses play a very important role in our lives. As advanced vet care and management strategies have improved, a larger number of horses are living longer, making this disease more common. Therefore, increasing our knowledge on the genetics underlying PPID has the potential to be extremely impactful to the entire equine industry. Identification of these potential disease-causing mutations will allow us to develop genetic testing to better screen and diagnose. This may be able to decrease the number of horses facing advanced disease and hopefully have less devastating consequences. Another exciting impact of this work may be its ability to cross species and increase the knowledge we have about Parkinson's disease in humans today and potentially even identify new and important mutations.